Our next podcast is going to go into cell division. So, what happens with the chromosomes when cells divide, and that's looking at what sometimes is referred to as the life cycle of the cell or the cell cycle. So, cells divide for a number of different reasons, and there's some certain behaviors that the chromosomes will take on during this process. So cells will divide for some different reasons. One of the main reasons is for growth. As you grow from an infant to a child to an adult, you have to add more cells to your body. And so that is growth. All living things grow. And so this is, in order to grow, we have to add cells, and the cells divide in order to get those new cells. And so this division is one of the functions of cell division. Cell division produces these new cells in growth and development. Another reason for cell division is for tissue renewal. So if you have injury to parts of your body and you have to replace those new tissues, and so that's a cell division can function in that. Also, in some organisms, single-celled organisms, when they divide from one cell into two cells, they are producing a new individual. They're producing an offspring. Or you can call it an offspring, but they're producing a clone of itself when it divides from one cell to two cells. So that's a form of reproduction, asexual reproduction. So here's an example. This is an amoeba here. It's dividing, it's finishing up its division from one cell into two cells, and then each cell will be an individual organism in this case. And so that's a means of cell division. So this means of cell division is a type of cell division we call mitosis. There's another form of cell division which is similar, but it only produces the sperm and egg. So it does not have these three functions, but rather it has a function of producing the gametes, and that's called meiosis. So we're going to be looking at both of those in this podcast. Okay, so in looking at the cells, we want to, in these processes of cell division, one thing that takes place is our genetic material behaves very consistently, and so that's important. In our somatic cells, which is all of our cells except for our sperm and egg cells, it's all of our diploid cells. We start with a pair of chromosomes in our nucleus. Now, the chromosomes are what the DNA, the genetic materials, is packaged in. So we can see this is a cell here. We can see the cell. We see the nucleus here in the middle, and they've let it absorb some dye so that you can see the chromatin here, which is the packages of DNA. Now, it hasn't completely formed into chromosomes just yet, but it is getting ready to. So it will form into chromosomes during this process of cell division. This will later package itself into specific packages called chromosomes, and this is a chromosome here. We'll learn a lot about these. Our whole unit that we'll be going into next deals with looking at the behavior of the DNA, which is what makes these chromosomes up, and what the structure and the function of the DNA that makes these chromosomes up. But here we're really just looking at the function of the chromosome itself in the process of cells dividing. So chromosomes are made up of two sections. The sections are called chromatids. So here we have one chromatid, and there we have another one. This shows the DNA molecule unwound, because it winds up very tightly to form these chromosomes. This shows it unwound, and so we have our two sister chromatids, which have a long strand of DNA wound together, and so they can split apart, and that's part of the function. So here has its one circled, so that's one chromatid right there. So we're going to start by looking at the process of mitosis, and then we'll go into meiosis. They're both have some similar phases and structures. So here we have, this is the kind of the life cycle here that we see of a cell. So here we have a cell, so we start with this cell right here. It goes through a normal growth phase, which is very long. So the cell will spend most of its life in this phase. This growth phase is called interphase. And so it spends a lot of time just in this interphase, growing along. And then the DNA starts to synthesize and come together, another growth phase here, and then all of a sudden, when it gets ready to divide, it's at the end of interphase it starts mitosis. So mitosis is, we're going to look at when all the chromosomes start to line up and split apart. So they're going to split apart in the middle. And so we have some phases that we can divide the process up in, and really these phases are dealing primarily with what is taking place with the chromosomes when this is happening. So we can divide it into, generally we divide it into 4 phases. Your book has thrown another one in here, and it's called the pro-metaphase. Generally it's just pro, meta, ana, tele. Your book has kind of an in-between phase that they've showed in here called pro-meta. And but the prophase is the first phase of mitosis, and then we move into metaphase, and then we have anaphase, and then we have telophase. All of these are about what's going on with the chromosomes. So here's a good diagram showing what's happening. Now in this diagram here, they have some electron microscopic views of a nucleus here with some bioluminescence to make the nucleus stand out, and the chromatin inside and the DNA and the chromosomes inside to stand out real brightly. And down here we have a figure where it's depicted of what's happening with the chromosomes, so you can understand better. So, as I was mentioning, the interphase is the long phase that the cell will spend most of its life in, and then when it gets ready to start mitosis is at the end of interphase, the G2 phase of interphase, and that's when all the chromatin is together, and then it begins to form into chromosomes. And so that will take place in prophase. It's the first phase of mitosis, and the chromosomes form. So that's one of the main things. And we also have the beginning of what's called the mitotic spindle, this yellow right in here and these 2 asters at the end. This mitotic spindle is going to form a web or network on which the chromosomes will hook to and help them to line up. And so that's prophase. Metaphase, skip over it, we'll come back to pro-meta. Metaphase is where they line up. So here our chromosomes line up in single file line right across the middle, our equator of the cell. The nucleus has kind of separated apart at that point. Now your book throws in this phase, which is an in-between called pro-meta, and that's where the chromosomes are getting ready to line up, and the nucleus is starting to disassemble. And so this is just where things are starting, but the metaphase is where they actually do it. So we have the chromosomes lining up. So in our cells have 46 chromosomes. So in our cells, they're going to line up single filely right across the middle of the cell. And then they split apart right in the middle in those chromatids. Remember, we just mentioned the sister chromatids that our chromosomes have. So there's two of them, they split apart right in the middle. So the next phase, anaphase, is where half of each of those chromosomes will go to one side and the other half will go to the other side of this cell. That's anaphase. And then telophase is where those halves form into a new nucleus, and so on each side of the cell, and that's going to be the new nuclei of our newly formed cells. And then it gets ready to split apart. That splitting apart is called cytokinesis, and that is where the cell splits apart. All right, so we can look at some other diagrams here. Here it shows a closer view. Here's a good electron microscopic view here of some chromosomes that are lining up, and you can see the microtubules that form part of that mitotic spindle that the chromosomes line up on. But here shows in this organism, whatever it is, has 1, 2, 3, 4, 5, has 6 chromosomes. So again, if we're looking at human cells, we'd be looking at 46 chromosomes that would line up across the middle. All right, and let's see. We can say also animal cells and plant cells carry out this same process when they get ready to divide. So when plants grow, they have to add more cells as well, and they go through the same process. The only thing that's different is when the cell gets ready to split apart, it's going to be different because plant cells have that hard, rigid cell wall. So in animal cells and human cells, the process is a folding in of the cell. Here's an electron microscopic view of a cell that's in the process of cleavage. And that's the process of the folding in of the cell, forming a cleavage here that is going to start the splitting of the cell, which is cytokinesis. All right, so we have this cleavage furrow that occurs in animal cells. Now in plant cells, it does not fold in like that. In plant cells, we have that hard, rigid cell wall. Here's our new plant cell and the nuclei have been formed from our one nucleus through mitosis. And now we're ready to form 2 cells. But rather than cleavage, we have the formation of what's going to be our new wall that will come in between the 2 nuclei. And this new wall is called a cell plate. And so that's cell plate formation in the plant cell. So that's a little different than in animal cells. So we can look at some of these. This is a good image here of a plant tissue here. You can tell because of the heart, the rigid cell walls here, but you can see that some of these cells are in different stages. Here we see an anaphase here where they've actually split apart. We see some metaphases where they're lined up. So that's a good picture there of some plant cells in the middle of cell division. So we can look at this. This is a diagram that shows that life cycle we just mentioned, but it also shows right here, right there during this process is when mitosis occurs. All that lining up of the chromosomes and the splitting apart, all of that's occurring here. So again, your cell spends most of its life in interphase. Okay, so we're going to end our podcast here, but we still have another type of division to go through, and that is meiosis, and we'll pick that up in our next podcast.